Yeah, my name is Marco van Zwetselaar. I'm calling in from Kilimanjaro, Tanzania. And this is going to be a little bit of a different talk from the previous ones. You know, when Greg Thane and I had an email exchange about the talk, he said, well, don't make it too technical. There's all kinds of people in the audience. And compared to the things I saw with millions of job submissions per week, well, that's not the level we're at. I'll glide you in easily to your break afterwards, while my family is waiting here for me to cook dinner. Anyway, so the talk came about when Todd on the mailing list asked people to drop pins on locations with condor pools, and I saw Africa was totally empty. And I could proudly drop 2 pins in Africa because I've set up 2 small HTCondor hpcs in 2 locations here. So this is going to be a talk a bit from the other side, both user and admin, because I'm sort of both with a group of people using it now, but we're not a big shared installation. But I'll tell you about how we got these HPCs running here, why they're running Condor, and how we benefit. So the 2 HPCs I've set up are at the Kilimanjaro Clinical Research Institute in Moshi, Tanzania. That's where I live. My wife and I moved here in 2012 when she became a doctor at the large hospital here, and I got a job as the IT guy at the research institute of the hospital, a clinical research institute of one of the big hospitals in Tanzania. I'm not a doctor or a medical researcher. Well, I'm not even a computer science guy. You know, I'm one of those who in the 80s started out on home computers and then never stopped programming. But a little bit more about the cluster later on. Then this year, I've set up another one in the Noguchi Memorial Institute of Medical Research in Ghana, in Accra. So most of my time I work for the Seek Africa project, which is a major UK aid grant to support labs around Africa in doing DNA sequencing for antimicrobial resistance surveillance and diagnostics. So it's a collaboration of the institutes that you would see here, plus Danish Technical University in Copenhagen. And well, you may have heard that you may have heard the problem of antibiotic resistance or antimicrobial resistance being the creeping pandemic or the silent pandemic. It was the cause of death for more than 1 million people in 2019, which is more than HIV and malaria combined. And it's estimated to be as big as cancer as the primary cause of death in 2050, with 10 million deaths a year. So there's quite a lot of research now going on into antimicrobial resistance. Our specific focus within the field is on the genomic epidemiology of AMR. That is, we study antimicrobial resistance on the genomic level. So the project is Fleming funded, which is a very large, well, UK investment in antimicrobial research around the world. Now, as I imagined an audience full of rocket scientists and physicists and so on, I thought I'd have to put in a little bit, you know, a 1-minute explanation of what molecular biology is all about and the stuff that we do. So here on the left, you see Francis Crick's central dogma, as he called it in 1958. He later realized that the word dogma wasn't appropriate, but yeah, that was years later. Anyway, at the top, DNA, the beautiful coil structure that sits in every cell of every living being. It consists of huge, very long sequences of 4 bases or nucleotides that we just abbreviate by their first letter, A, T, C, and G. So in terms of the work that we do, that is our data. We deal with huge databases and files full of A, C, Ts, and Gs. DNA carries the genetic information that gets replicated between generations, and meaningful stretches on the DNA we would call RNA. RNA gets transcribed via all sorts of complex processes from the DNA, and then gets translated into proteins. And the proteins are the things that make up biological structure and function. So that's sort of molecular biology in 1 minute. The relevance to antimicrobial resistance and the sort of studies that we are doing is you could imagine DNA coding for a protein, an enzyme, that breaks down the active compounds in an antibiotic, or developing a mutation that would make the antibiotic not catch on to the genome to destroy, well, eventually the bacteria. That's where we are. That's sort of the area where we are working. Even in the 3 talks before this, I was counting the number of times that I heard the word knob, and I just realized one thing. So, in terms of terminology, biologists have a gift for doing neologisms. So, if you hear a biologist use the word that ends in "ome," like genome, what they mean is it's all the genes together. So you have one gene, you have a genome. You have a lot of transcribed genes. Well, we call that a transcriptome. You have translated proteins, that would be the proteome. And so I was thinking, you know, as a thing to pick up for Condor, the knobome would definitely describe all these knobs that Condor has, and then knobomics would be our shared struggle to understand them in their entirety. But that's a sideline. So what we do in our studies is we take DNA from bacteria, we sample from infections in humans, for instance, and then try to read that DNA into data files, the thing that we deal with. And what this slide shows is that over the course of time, since 2003, when the first human genome was sequenced, sequencers have become incredibly powerful, initially just sequencing about 100 million bases at a time with the Sanger sequencing. That's when the first human genome was sequenced, to several terabytes per run, as we call this, of the sequencer at the top. The other development in sequencing has been that sequencers are able to read longer and longer fragments of DNA through time. So in the green circle are the sort of sequences that we would be using for microbial sequencing nowadays. So they're called benchtop second generation sequencers, and they get about 25 to 250 gigs per run of data. The MinION is one of the most interesting developments of the last 10 years. It's a portable device, the size of a USB stick, that you actually stick in the side of your laptop, and that you can take into the field to sequence DNA. And it also yields in the order of 20 gigs of data per run. So clearly, we're not looking at the scale of other projects that I've seen presenting here. But for microbial genomics, and in particular, of course, once you move to human genomics, the human genome is about 1,000 times bigger than a bacterium. One human genome is 3 billion reads. It's 3 billion bases. Bacterium would be 1 million to 6 million or so. That's the sort of data that we're dealing with. So with the biology behind us, so what's the work of what's the job of bioinformaticians? We get the genomic data and try to distill that into useful information. One of the first things that we would do is sequencers produce small fragments of DNA when they do that run. So we end up with, at the end of a sequencer run, hundreds of millions, up to 1 billion or so of small fragments of DNA, and we want to reconstruct that into a genome. That job is called assembly, and it has been likened to, you know, putting a pile of a few hundred newspapers on top of each other. Today's edition of a newspaper, blowing them up into tiny shreds, hundreds of millions of shreds, and then trying to puzzle that back together. So this is the sort of compute job that you would start with. Other things that we would do in genomics and bioinformatics is looking for antimicrobial resistance genes. We wouldn't need to reconstruct the whole genome of the organism. We might just take the source reads, the stuff that comes from the sequencer, and try to map that on a reference database that has all the known antimicrobial resistance genes or virulence genes, or, you know, genes coding for other sorts of things. So that's two of the kinds of compute jobs that we do. Another one, which will have been, well, I would say familiar to anyone over the past COVID-19 pandemic, it's the construction of phylogeny. In other words, you take thousands or hundreds of thousands of genomes of, for instance, in this case, the SARS-CoV-2 virus, try to compute a large matrix of their mutual similarities, and then try to reconstruct a sort of a family tree of where they came from, in order to identify variants that would be of public health concern. For instance, everyone will have heard about the infamous spike gene on the SARS-CoV-2 genome. So that's some of the things we do in bioinformatics. But of course, you know, the question was, how did the Condor land on the Kilimanjaro? This is the current server park at Keisharai. It may look tiny to many of you, but and I'll let you guess the names of our servers from the who is who pictures. And the one at the right bottom is the one where it all started with. So in 2013, we won a grant together with Danish Technical University to set up a sequencing facility in the Keisharai lab in Moshi, Kilimanjaro. That was before pretty much anyone was doing much bioinformatics or genomics here. And what we started out with were 3 laptops, pretty beefy for 2013, and they had the pipeline that Danish Technical University used for all their microbial genomic analyses. It was a port of the software that ran on their big cluster, which was aptly called the computer roam, of course. And it was ported to these laptops. But even though the system that was on it was called PBS, the portable batch system, it didn't very much work. So I started to work on an alternative. I looked at Slurm, then of course I looked at Condor, and Condor had this marvelous feature that we could use brilliantly, because we had 3 of these laptops. One was had the ported PBS. The other two were used by me and my colleague. What Condor could do was it could run on the three tied together, and then do its work while we were off to do other things. That set it apart from PBS and Slurm in those days. I must admit there was another reason for us to choose Condor, which was that when I read that the LIGO project was using it, I mean, the coolest physics experiment since Galileo was dropping rocks off the Tower of Pisa. So that's how I got to install Condor on it. And over time, since 2013, I managed to get multiple grants to build it out into a 4-node cluster with one large 2 terabyte memory node, half a petabyte of storage in an offsite backup, 2 GPU nodes. And just as an aspect of what makes this typically Africa, we did not have the network capacity to do all kinds of things online. Even now, when I need to download a 25 gig database, that is a day, 2 days download. So it really is intended for local deployment. And I think as the final, and actually this brought down, weekend long runs on 1 or 2 laptops to, by now with the 4-node cluster down to a run that takes about 1 hour before we hand the data to our customers, to the researchers that send their isolates. Finally, I think as a huge what do you call it, like a huge benefit of using Condor in our setting is, I don't know how this is in other fields, but in genomics and bioinformatics, the word is pipelines. So there's hundreds, thousands of tools being developed all the time in bioinformatics, some very generic, others very dedicated to certain purposes. And much of our work consists of tying them up into pipelines that do useful work. So you see the colored boxes here would be all these tools that I sort of linked together into what would be called a pipeline. And this diagram shows our standard bacterial analysis pipeline. So we would take, one run of 100 samples, 1,000 samples, or any number of samples, throw it in, and at the end have some tables to give to the customers, plus a whole lot of analysis data that they want. Of course, to anyone who knows a bit of Condor, this diagram just screams, "Dag, man!" and yeah, it was a bit of a struggle initially to get that working, but now that it's running, the big benefit is what used to take us days of hands-on time to set up the analysis for a bunch of samples is down to 10 minutes of setting up an input sheet. Let's Condor scatter the work over the CPUs and the nodes, and then an hour later we will have all the data, compared to literally a week or more of work. So here's a big thank you to the Condor developers from Africa. We're happily using it, maybe totally different from what I saw in the previous 3 presentations, but highly valuable and much appreciated. Thank you so much for your attention.